Welcome to this explainer. Okay, so if your biology education ended in high school, you probably picture mitochondria as these simple, static little jelly beans that just pump out energy, right? Well, scratch that. Today, we're going to completely redefine everything you thought you knew about the so-called powerhouse of the cell. As you'll see, they are actually dynamic, shape-shifting master regulators of life, disease, and, incredibly, even cellular death.
Let's dive right in by throwing out those boring textbook diagrams and asking a question that sounds like straight-up science fiction but is actually a biological fact. What if the powerhouse of your cell is actually an ancient bacteria? I know, it sounds totally crazy, but to understand how these organelles control our health and our aging, we really have to start at their deeply weird origins.
We've got a lot of fascinating ground to cover in this explainer. We'll be moving beyond the cellular powerhouse, looking at the ATP energy factory, and exploring their role as regulators of life and death. Then, we'll get into shape-shifting and quality control, disease, aging, and cancer, before wrapping up with the future of medicine. So, let's get started.
Section 1. Beyond the cellular powerhouse. Let's start with this origin story, because it brilliantly illustrates why mitochondria are just so utterly unique compared to everything else inside our bodies.
So, the modern updated definition of mitochondria is far more complex than just a simple battery. Yes, they are specialized double-membrane organelles responsible for energy production, but crucially, they also handle metabolic regulation and cellular signaling. You'll find them in almost all eukaryotic cells, and they pack in massive numbers inside tissues that demand the most energy. Think your muscle cells, your neurons, your heart, and your liver. But here's the thing. They aren't originally from around here. Enter the endosymbiotic theory. This proposes that mitochondria actually originated from ancient free-living bacteria, specifically related to alpha proteobacteria, that were literally swallowed by early eukaryotic cells billions of years ago. But instead of being digested, they formed a partnership. And the evidence for this is absolutely overwhelming. I mean, they contain their own DNA. They have bacterial-like ribosomes. They reproduce by dividing through binary fission, just like bacteria do. And their membranes perfectly resemble bacterial membranes. They are essentially a microscopic organism living right inside of you.
And the crucial point here is that they still operate with a remarkable degree of independence. To put that into perspective, the human mitochondrial genome contains exactly 16,569 base pairs. This circular DNA encodes 13 essential proteins, 22 tRNAs, and 2 rRNAs. Oh, and here is the kicker. This genome is almost exclusively inherited maternally. You got your entire mitochondrial blueprint directly from your mother. So, thanks, Mom.
Section 2. The ATP energy factory. All right, let's move to the main event most people remember from biology class and see exactly how this microscopic engine actually generates the energy that keeps you alive. Think of cellular respiration as an incredibly elegant factory assembly line. It all starts out in the cytoplasm with glycolysis, breaking glucose down into pyruvate. Then the raw materials move inside the mitochondria. Stage 2 is pyruvate oxidation in the mitochondrial matrix, converting that pyruvate to acetyl-CoA. Next up, stage 3 is the famous Krebs cycle, also spinning in the matrix, generating carrier molecules like NADH and FADH2. After that, the assembly line shifts to the inner mitochondrial membrane for the electron transport chain. Here, a series of complexes transfer electrons and pump protons across the membrane to build pressure. And finally, the grand finale, oxidative phosphorylation. An enzyme called ATP synthase acts exactly like a microscopic water wheel, using that proton gradient to literally spin and produce ATP. This beautiful 5-step assembly line progressively breaks down glucose to generate the vast majority of your cellular energy. Section 3, regulators of life and death. But, producing all that energy has consequences. It turns out mitochondria don't just give life. They hold the absolute ultimate power over a cell's survival. During that massive ATP production process we just looked at, mitochondria generate exhaust in the form of reactive oxygen species, or ROS, like superoxide and hydrogen peroxide. It's a really delicate balance. Low levels of ROS are actually good. They are actively used for cell signaling and immune responses. But excess ROS, that's bad news. It becomes incredibly destructive, causing oxidative stress, DNA damage, and ultimately mitochondrial dysfunction. And this brilliantly illustrates their darker role. When things go terribly wrong, mitochondria act as the cellular Grim Reaper through a process called apoptosis, which is programmed cell death. It's regulated by specific proteins. BCL2 tries to stop it, while BAX and BAC try to trigger it. A damaged mitochondrion will literally release a protein called cytochrome C. This activates executioner enzymes called caspases, essentially pulling the plug and orchestrating the complete destruction of the cell from the inside out. Wild, right? Section 4, shape-shifting and quality control. With stakes that high, the cell obviously needs a reliable way to keep these power plants in perfect working order. Remember how I said earlier they aren't static jelly beans? Well, mitochondria constantly change shape through a process called mitochondrial dynamics. Through fusion, which is driven by proteins like mitofusin and OPA1, two mitochondria actually combine to pool their resources, exchange components, and improve their overall function. On the flip side, through fission, driven by a protein called DRP1, one mitochondrion divides into smaller units. This is crucial for distribution, but more importantly, it lets the network isolate and chop off a severely damaged section. So once a damaged section is chopped off, how does the cell clean up the mess? Enter mitophagy. This is the selective removal of damaged mitochondria. When a mitochondrion is beyond repair, proteins like PINK1 and parkin act like a biological tagging system. They flag the defective unit for destruction. This vital quality control process maintains our cellular energy balance. In fact, defects in this exact PINK1 and parkin system are heavily associated with Parkinson's disease, which just shows how absolutely critical this cleanup really is. Section 5, Disease, Aging, and Cancer. Now that we understand the micro-level mechanics, from energy production to quality control, let's scale up and see what happens to the human body when these incredibly complex systems begin to fail. Because tissues like muscles and neurons in the heart demand so much energy, inherited mutations in mitochondrial DNA are absolutely devastating. When mitochondrial DNA mutates, conditions like Mella syndrome can cause life-altering strokes and lactic acidosis. If mutations affect cellular respiration, diseases like LHON can actually lead to sudden vision loss. And if ATP production is generally defective, you get mitochondrial myopathy, leading to severe systemic muscle weakness. What's worse, as we age, our mitochondrial DNA naturally accumulates these mutations. ATP production drops, destructive ROS spikes, and that mitophagy cleanup slows down. Essentially, our cellular batteries just wear out over time. But what's truly sinister is how cancer cells hijack this exact biology through something called the Warburg effect. Normal cells use mitochondria to make energy efficiently with oxygen. But cancer cells, they preferentially alter their metabolism to use glycolysis, the less efficient first step of energy production, even when oxygen is fully available. Why on earth would they do that? Because these metabolic changes allow cancer cells to grow incredibly rapidly, adapt to high stress, and, most importantly, actively evade that apoptosis death signal we discussed earlier. They literally turn off the Grim Reaper so they can multiply unchecked. Section 6, the future of medicine. The good news, we are no longer just passively observing these processes under microscopes, we are actively learning how to hack them. Biotechnology is advancing at lightning speed right now. The frontier of research now includes using AI-based prediction and mitochondrial gene editing to actually repair mitochondrial DNA directly. Scientists are developing mitochondrial replacement therapies for inherited disorders, synthesizing artificial mitochondria, and creating targeted drugs for neurodegeneration. There's even massive funding pouring into mitophagy enhancement and NAD+ metabolism repair to literally slow down the cellular process of human aging. It's a real game changer. Before we wrap up, it's just incredible to see how every structural component we've discussed today is purpose-built. The outer membrane controls movement in and out. The folded cristae of the inner membrane house the electron transport chain and that ATP synthase water wheel. The matrix safely holds the DNA and Krebs cycle enzymes. And proteins like PG1-alpha regulate the birth of brand new mitochondria when your body demands more energy, like, say, during a heavy workout. Form perfectly, absolutely matches function. So, I leave you with this final thought. We've seen today that mitochondria are so much more than just a powerhouse. They're dynamic, shape-shifting networks that govern our metabolism, signal our immune system, and orchestrate our cellular death. Considering these ancient, engulfed bacteria hold the ultimate blueprints to human energy, inherited disease, and the aging process itself, if mitochondria control how our cells die, could mastering them be the ultimate key to extending human life? Think about that. And thank you for joining me in this explainer.